Assalamu alaikum viewers. Today is the second video of general embryology, which is the spermatogenesis. In first video, we already discussed about gametogenesis. In this video, we will discuss about spermatogenesis in detail. In 3 slides, we will cover the whole spermatogenesis. Now, first, what the word spermatogenesis mean? Spermato means sperm, genesis mean formation. Mean the process of formation of sperm is called spermatogenesis. How we define spermatogenesis? It is the process of formation of sperm in the seminiferous tubule of testes. Now, for sperm, for spermatogenesis, sperm, you should remember 3S. S for sperm or spermatogenesis, and spermatogenesis occur in second S, which is the seminiferous tubule. The third, it is supported by third S, which is the Sertoli cell. Mean spermatogenesis, for spermatogenesis you should remember 3S. S means spermatogenesis or sperm. Second, it formation occur in the wall of seminiferous tubule. Third S, it is supported by Sertoli cell. And next is the interstitial cell, which produce testosterone, not sperm. Now, after definition, come to the aim of spermatogenesis. What are the aim of spermatogenesis? There are 3 aim. Number 1, a reduction of the number of chromosome from diploid 46 to haploid number 23 by meiosis. This is the first aim of spermatogenesis. Second aim is the change in the shape of the male germ cell to produce a highly motile sperm ready for fertilization of the ovum. Mean the second aim is to change the shape of the male germ cell to become a highly motile sperm to ready for fertilization of the ovum. Third aim is the increase in the number of cells so that one mother cell means spermatogonium, produce 8 sperm cells, mean to produce high amount of sperm cells. These are the 3 aims of the spermatogenesis. Now come to the occurrence. Where spermatogenesis occur? Spermatogenesis occur continuously in the seminiferous tubule of testis. Mean the occurrence is in the seminiferous tubule and starting from puberty till old age. Now come to the stages of spermatogenesis. There are 2 stages of spermatogenesis. Number 1 is the spermatocytogenesis and second is the spermiogenesis. What is spermatocytogenesis? In spermatocytogenesis, from spermatogonia cell, germ cell, spermatids are produced. And in second stage, spermiogenesis, spermatids are converted into mature motile sperm. Now, first the first stage is the spermatocytogenesis, in which from germ cell the spermatid are produced. It is a process by which the spermatogonia differentiate into spermatid as follows. Now, in spermatids, look at the left side of diagram. First, from spermatogonia, with the help of mitosis, there will be daughter spermatogonia. Then daughter spermatogonia, again due to mitotic division, form primary spermatocyte. Then primary spermatocyte divide by meiosis, give secondary spermatocyte. Give 2 secondary spermatocyte. Then these 2 secondary spermatocyte divide with the help of mitosis to form each secondary spermatocyte produce 4 spermatids. By mitosis, in the result there will be 8 spermatids. 8 spermatids. Now, in spermatocytogenesis, it is a process by which the spermatogonia differentiate into spermatid as follows. Number 1, The spermatogonia are the most primitive male germ cells. They contain the diploid number of chromosomes, 44 autosome plus 2 sex chromosomes, X and Y. This lie in the wall of seminiferous tubules supported by Sertoli cell. Each spermatogonium undergo mitotic division to give rise to daughter. Each spermatogonium undergo mitosis division to give two daughter spermatogonia, each of which contain 44 autosome plus XY chromosome. Each daughter spermatogonium grow to give primary spermatocyte, again by mitosis. Then the primary spermatocyte undergo meiotic division to give rise to secondary spermatocyte. Each of contain the haploid number of chromosome, 22 autosome plus X or Y sex chromosome. Then each secondary spermatocyte undergo mitosis to give two spermatid. Thus, four spermatid are formed from each daughter spermatogonium. In the result, there will be 8 spermatids. Now come to the second stage, spermiogenesis. Now the spermatids converted into mature motile sperm. This is called spermiogenesis. The spermatids, which is a rounded cell, undergo metamorphosis. Metamorphosis means morphological and structural changes to become transformed into mature sperm. Now, in metamorphosis, the morphological and structural changes are 5 in number, from which spermatid change into sperm. Number 1, the nucleus become condensed and form most of the sperm head. Second, Golgi apparatus form the head cap. Golgi apparatus form the head cap covering the interior half of the nucleus. Third, the centriole elongate to form axial filament which transverse divide the neck, middle piece and tail of the sperm. Fourth, the mitochondria form a spiral sheath around the middle piece. And finally, the remainder of the cytoplasm is shed. Only nucleus take part in zygote. And in zygote, from mother, both nucleus and cytoplasm come. But from father, only nucleus of sperm come, not cytoplasm, because cytoplasm become shut. This is the spermiogenesis. Now come to the mature sperm. The size of the mature sperm is 16 micron long. Structure, it is formed by following parts, 4 parts. Number 1 is the head. The head of the sperm, mature sperm, is 5 micron long. It is formed mainly by nucleus of spermatid and carry the genetic information. And its head is found mainly by nucleus of spermatid and carry genetic information. Its interior, look at the diagram and the lip. Its interior half is covered by a chromosomal cape which contain a chromosomal enzyme to help penetration of the ovum. Second part is the neck. Neck is the very short and contain 2 centriole. In neck there is a 2 centriole and very short part. Third is the middle piece, which is 5 micron long, formed by axial filament surrounded by mitochondrial sheath. Why mitochondrial sheath covered? Concerned with energy production. And fourth part is the tail, which is 50 micron long, formed of axial filament, same like middle piece, but here it is not covered by mitochondrial membrane, it is covered by thin protoplasmic membrane, and is concerned with mobility of the sperm. Middle piece with energy production, tail motility of sperm, head genetic information, a chromosomal enzyme. Now come to the transport and fate of the sperm. Transport, where the sperm become transport after seminiferous tubule, and next is the fate of the sperm. In testes, after seminiferous tubule, the sperm come to epididymis for physical maturation. After that, it go to vas deferens for storage, and in vas deferens, they are ready for ejaculation. If there is no ejaculation, they become dead and recycle. Now the sperm leave the testes to reach the epididymis for physical maturation. Then they reach the vas deferens, waiting for ejaculation. In vas deferens, it is in dilated end, which is known as ampulla of the veins. When ejaculation occurs, the sperm pass through the urethra, where they become deposited in the vagina, ascend through the cervix, and reach to the cavity of the uterus, enter the fallopian tubule to reach its lateral part, ampulla, for fertilization of egg. If no ovum is found in the fallopian tubule ampulla part at the time of ejaculation, the sperm remain for 96 hours, then die. The fertilization power of the sperm is lost about 42 hours after ejaculation. Remember, in female fallopian tube, if there is no ovulation, sperm remain for 96 hours, then die. The fertilization power of sperm is lost about 42 hours after ejaculation. If no ejaculation occurs, the stored sperm inside the vas deferens die and become absorbed. Next is the seminal fluid, the semen. Its nature, characteristics, volume, number, mobility, and abnormality. Now, first is the nature of the seminal fluid or semen. When male in result of ejaculation, the semen out which contains the sperm. Nature, it is the fluid containing the sperm suspended in the seminal fluid actually is the secretion of the seminal vesicle, prostate gland, and the bulbourethral gland. In this fluid, the sperm is present or suspended. Now, characteristics, it is thick, white, opaque, gelatinous fluid. It is a characteristics odor and is alkaline in reaction and also alkaline in nature. The third is the volume, 3 to 5 cc per ejaculation. Its volume normally not less than 1 to 5 cc. 1.5 cc. Mean its volume is from 3 to 5 cc per ejaculation. Normally it should not less than 1.5 cc. Next is the number of sperm. Per ejaculation there is a 200 to 300 million of sperm in per ejaculation in seminal fluid or semen. Next, motility, normally 60 to 70% of all sperm are motile. Next is the abnormality. There are three types of abnormality in the of sperm in the semen or seminal fluid. Number one is the abnormality in shape. Second is the abnormality in number. And third is that there is a presence of dead sperm in the semen. First is the abnormality in shape. The abnormality may affect the head or tail. The sperm may be too small, which is dwarf, or too big, which is giant, with the two head bicephalic, or with two tail bicaudic. Normally, the abnormal form of sperm in the semen does not exceed 10% of the total sperm count. Mean the abnormal form do not exceed 10% of the total sperm count. If more than 25% of the total sperm count, the abnormal, then the fertility will be impaired. Next is the abnormality in number. In number, if there is a number one is the oligospermia, oligo means few. The number of sperm is below normal. This is the oligospermia. Azoospermia is the complete absence of sperm in the semen. If there is a complete absence of sperm in the semen, there is no sperm in the semen, it is known as azoospermia. And if there is a below normal number of sperm, this is known as oligospermia. The third is the necrospermia. Necro matlab just like necrosis, dead. Necrosis is the presence of dead sperm in the semen is known as necrospermia. Now, today this is all about spermatogenesis. In coming videos of general embryology, we will discuss about oogenesis, then weeks of development. Now, On my YouTube channel, there is more than 550 videos are present of different subjects of MBBS and BDS subjects. Now, if you like my video, press the like button and share this video with your friends. If you are new to my channel, subscribe my channel by pressing the red subscribe button and also press the bell icon in order to receive my each new video notification. For watching my videos, I am very thankful to you. Thank you very much.